So we are going to talk about star codons other than AUG which are present. So in both eukaryotes and prokaryotes, there are alternative star codons present, and when translation happens to these star codons, the translation efficiency in eukaryotes will be much lesser than in prokaryotes, because in prokaryotes the ribosome directly binds to the strand algorithm sequence, whereas in eukaryote the ribosome has to scan the mRNA to find the start codon sequence. So it might miss the alternative start codon which is present. Now the lock and key structure between the initiator anticodon and the alternative start codon is weaker. So the translation process will happen at a slower rate, because initiation is the rate determining step. And we know that a lot of translation happens simultaneously in a single mRNA. So there will be some ribosomes which will bind to the AUG start codon, and some which will bind to the non-AUG start codon. So there will be different forms of proteins which will exist, which are called isoforms of proteins. Now when these alternative start codons are there, they are created due to a vowel at the first position of the codon. Like instead of AUG, it is UUG or CUG or GUG. So like instead of the vowel-based pairing, which happens at the third position, this is at the first position which happens. So I will pass over to.
So GUG usually codes for value. It's a near cognate codon which translates with a lower efficiency as compared to that of AUG. So it is the most common alternative of AUG. At the start of the sequence, it recruits for the methionine and for N-formyl methionine in prokaryotes. Often it produces only 12 to 15% of protein levels as compared to the standard start codon AUG. For example, in the E. coli bacteria, it produces approximately 14% as opposed to that of the standard start codon, 83% of proteins. What are its uses? There are two main uses. One is the regulatory usage and the other is in the human mitochondria and the viral genome. The regulatory usage, when it comes to E. coli bacteria, is in the LAC-I repressor gene, where it reduces, limits the production of the proteins. And in human mitochondria, it helps in the translation of the ATP6 producing gene, which is necessary for the production of energy. It's also used in the viral genome. The next part will be continued by Karthik.
So now UUG as a start codon, and UUG as a start codon is found in prokaryotes, and as a start codon it codes for methionine just like AUG. And comparing to GUG, AUG is thermodynamically less favorable because now it has to match to the anticodon UAC. Which is less favorable, and this energy gap helps the protein helps in the regulation of protein production. Like some specific protein has to be produced limitedly, and this UUG as a start codon, this helps the cell to produce the protein in limit. And cells uses the mechanism like UT scanning. This offers proteome diversity, and this will be discussed in detail by Jivika in the next slide. And one of the examples I would like to include here is like when cell is introduced to some any stress response or anything, Like it will change, it will use this leaky scanning mechanism, and it will produce different protein in different conditions by using the same gene coding. And this study is widely used in research, particularly in experimental biology and synthetic biology. I would now Jirika would continue this.
So hi, now I'm going to talk about CUG start codon. It is mostly common in eukaryotes. It uses a method called leaky scanning where normally we see the 40S ribosomal subunit start scanning and it attaches to the 5' end of the mRNA sequence. It scans until the first start signal, but here in leaky scanning, the ribosome leaks past the first site and it continues elsewhere. So in the case of COG, it continues in the downstream so that it can produce multiple different proteins from a single mRNA sequence. So this is a proof that genetic code is not a rigid blueprint, but it can be a flexible system. By starting translation at COG, we can develop the protein function without needing extra genes. As a collective sequence, like biological significance of these start codons are protein diversity, stress response, and we can even take it to the evolutionary edge. And yeah, thank you.
Okay. Yeah. So, any other question? Yes, you may.
So this process can it happen in all proteins or only in specific proteins?
Then it actually depends on the condition.